Thank you for the introduction, and thank you for this opportunity to give the results to this inter-laboratory exercise that we carried out. So my name's Claire Browning and I work for the Pirbright Institute. This study is to evaluate the use of 2 commercially available NSP kits, and obviously these ELISAs are used in post-outbreak surveillance to prove freedom from disease with or without vaccination. Okay, so the study plan was to look at the 2 commercially available kits. We chose the ID.Vet screen, ID.Vet ELISA, which you've just heard from Loic, and the PrioCHECK NSP ELISA. ID.Vet offer the single and overnight protocol. So with that in mind, we looked at Emmy's previous study, which obviously looked at 6 ELISAs. This is obviously only looking at 2 NSPs. Hers had a lot more samples, but we have covered the same species, so cattle, pigs, and sheep. Okay, so this was a study that we decided to select 5 laboratories, all that were ISO 17005 accredited, and then within each European laboratory we also had 2 operators that were 17005 accredited as well. So each operator was sent a blinded randomized panel consisting of 180 samples. So we took, from the 3 species, we have 30 negative and 30 positive cattle, the same for sheep, the same for pig. The only thing I'd like to point out is the lack of true NSP positives when it comes to porcine samples. So we did have to use one of the stronger NSPs and titrate out so that we could mimic a range of NSP values for those. So as I said before, each operator would then run the assay, so the PrioCHECK, the ID.Vet short, and then ID.Vet long, and all samples in the panel were tested in duplicate. So each laboratory received 360 samples. So the samples we decided to include in the panel, we were looking at more the clinical status of the animal. So we selected experimental serum from animals that were a minimum of 8 days post-infection, whether this was vaccinated, infected, or non-vaccinated and infected animals. Each sample was also BI inactivated and acuity tested so that it was safe to handle in a lower containment laboratory. And the negative serum that we selected were from FMDV-free countries. So each operator was tested their samples, which is a lot of data when you consider that we asked them to test it in duplicate. So we've done a comparison, so for all 5 labs, with the 10 operators, and this data is showing the comparison of along the x-axis is the PrioCHECK NSP, and the y-axis was the ID.Vet short ELISA. As you can see, they're clearly distinct areas where these are your positive samples, and down here are your negatives. And in the middle, where both kits have that 50% cutoff, whether it's measuring PI or SN ratio, we looked and found that there was a 95.5% concordance between the animals, which is what we hopefully expected for the study, because it's not looking at which kit is better, it's more having more kits on scope to be able to be interchanged during an outbreak. So these kits do compare very well. The one thing the study did bring up for us when looking at the data, we had 6 negative samples that were from FMD-free countries that actually had very high NSP values. So this was something that we weren't expecting. So each lab, each operator across all 3 tests gave the same positive NSP result. So this, we then decided to carry out further evaluation of these samples. So we looked at what could we reproduce this false positive result. So we had 3 experiments where we looked at the heat and activation process. So for us, we heat and activate at 56 for 30 minutes. We titrated the positive samples and true positives, as well as the samples that gave us false positives. And then we tried to replicate if we could reproduce the positivity, leaving samples out at room temperature for 72 hours. So there's 6 samples, and then for each sample, we had 4 aliquots. So we took from just the Pirbright, operator 1, operator 2 aliquots that were provided in the panel. We then took the inside stock that we did preliminary data with, and then we took a fresh outside stock that had been used for the study. So looking at heat and activation first, you can clearly see each colour on the screen. So here, this is one sample, 2, 3, 4. So the darker coloured blue, for instance, this is aliquot 1, aliquot 2, before and then after heat and activation. So the 2 component operator samples this, and the other thing I need to point out is these were only carried out on the PrioCHECK ELISA, this extra data that we did. So before heat and activation, we had a PI of over 95, which is clearly very high, seeing that it's 50 cutoff. And then after heat and activation, the PI value dropped considerably. The only exception was this ovine sample, so we're not quite sure, but you can clearly see that heat and activation does play a role in your PI values. The next thing we did was to titrate out the non-heat and activated aliquot. So up here I have a positive bovine, ovine and porcine sample. So we did a simple dilution series, and as you can see that they taper off quite steadily, showing that they are true positives. But when we titrated out the false positives, we saw this where it basically just drops straight off or carries on a little bit and then drops. So clearly there's distinct curve happening, so indicating that these are false positive samples. Okay, so then the third part was to look at if we could replicate this issue with storage conditions of samples over long term. So as I said, we took a fresh aliquot. Each of these aliquots was separately aliquoted. So we left one at room temperatures for the 72 hours and then one in the fridge. All the tests that we carried out, we tested on the same day to make it as consistent as possible. So most of the results didn't really show much of an increase except for this one sample, which is obviously an indication that this can happen. I'm not saying it's a total, this will happen, but there is something to bear in mind with these false positive results when you're testing your samples. So in conclusion, the objective of the study was we've proved that the data supports the idea that 2 commercial assays have equivalent performance for the detection of FMDV NSP specific antibodies. And it provides concordance and validation data to accredit alternative assays for routine diagnostic purposes, hopefully mitigating any potential supply issues that could arise during an outbreak, especially if there's more than one outbreak in Europe. There's going to be a sudden demand for more kits. So the more kits you kind of have in your backup plan, probably the better, so that you can interchange them. And also the study, as I've just mentioned, also highlighted storage and heat and activation of maybe serum samples, as it plays an important role in non-specific binding. And this obviously isn't an isolated incident. I know Labib at ANSES has seen similar issues. But the other thing is in an outbreak situation, you will be processing your samples fairly rapidly, so you won't probably have this inactivation, heat and activation issue where you need to, but we routinely heat and activate any samples we test anyway. But it's something to bear in mind with post-vaccinational studies that are carried out in other countries. If you're going to collect all your samples before shipment and testing, that kind of thing may be something to bear in mind so that you know when you're testing. And I'd just like to thank EuFMD and also Thermo Fisher ID.Vet for providing the kits, and all the laboratories for participating in this project. Thank you. Thank you.